In this video, we're going to begin talking about the history of microbiology. So let's get started. So we know from the writings of many ancient cultures that there were rules that were meant to promote sanitation and to protect the health of the people in those cultures. And so, for instance, in many cultures, there are taboos against eating pork, because pork is a carrier of parasites, and if you eat poorly prepared pork, then you risk getting those parasites yourself. And there were rules where people were, gathered together. There were rules about what can and should be done with fecal material in order to prevent those fecal oral or fecal to other modes of entry into the body transmission of disease. And people understood that clothing and that objects like bedding or even objects that were, especially those that could absorb fluids, were capable of transmitting diseases. And so, I got this picture here of bedding and clothing being burned in order to prevent the transmission of disease from these materials. But it wasn't until the mid-1600s that the first microscope was built, and that people could really lay eyes on, or at least the first known microscope was built, and that we could lay eyes on on the small microbes that were found in fluids, or even see things as small as cells. Robert Hooke built the first compound microscope. It had 2 lenses in it, and that's what we mean when we say compound microscope. It is a microscope that has 2 or more lenses or sets of lenses in its construction. And he looked first at really thin slices through cork. Cork is a tree bark product, and so it is built of cells. And he observed these tiny connected boxes that reminded him of the rooms in which monks lived. They're called cells, and so he named them cells. And it was he who first deduced that cells are the smallest living structural unit of life. And so here's a drawing from Hooke's own book that he wrote, Micrographia, it was called, where he shows his system for looking at things under the microscope and some of the drawings that he made. And so here's the cork looking at the cells that he saw in cork. In the late 1600s, Anton van Leeuwenhoek, or Antony van Leeuwenhoek, he was a Dutch cloth merchant. He began building very high-quality microscopes. They had the capacity to magnify up to 300 times, usual or visual naked eye size. And he just went crazy looking at stuff. He looked at the goo that developed between his teeth. He looked at his own semen and found sperm. So anything that he could think of to look at, he would do it. He was the first to describe sort of the breadth of microscopic life. He drew pictures and described free-living algae and protozoans. He saw the 3 different shapes of bacteria that are the most common shapes that we now call cocci, bacilli, and spirilli. And he looked at fungi and began drawing and identifying fungi as well. He was really, really particular about his microscopes, and he kept secret his process for producing such high-quality lenses. He wouldn't sell them, and so after he died in 1723, there were very, very few advances, really, in microbiology, because nobody had access to the technology van Leeuwenhoek had developed in order to build these high-quality microscopes. And here we're looking at a replica of one of the microscopes that he built. The little pointy part, so if we're looking here. Let's see, it's probably easier. Yeah. So here is where the lens is, and he would mount a little bubble of fluid with whatever the object that he was looking at there, right on the tip of that little point, and capture it kind of between the point and the lens that's in there, and he could adjust it, kind of move it up and down, and that was how he peered through that eyepiece, and so that was how he could look at all of these things. And it was quite a loss when he died without sharing the technology that he had developed. So we take a big jump now into the 1830s, and two scientists, one a botanist and the other a zoologist. They were both Germans. Their names were Matthias Schleiden and Theodor Schwann. So Schleiden and Schwann were the ones who formulated or clearly articulated what has become known now as the cell theory, the idea that cells are the fundamental unit of life, and they carry out all of the basic functions of living organisms. So they looked at a bunch of things and they realized that, hey, everything is built of cells. And so we now accept this as a theory, and we're going to, in just a minute, I'm going to talk a little bit about what theory means. For a biologist, a theory, or talking about a theory is similar to a physicist talking about a law, like the law of gravity. Theories are widely held to be explanatory of the natural world. And so the cell theory applies to all living organisms that we've discovered so far, so we feel pretty good that it applies to all living organisms, but not to viruses. Viruses are, it is moderately debatable, but I suppose most people, or I believe most people, do not consider viruses to be living organisms. So we'll take a brief aside here to talk a little bit about scientific theories and hypotheses. We have a tendency to say, well, I got a theory about, whatever thing you noticed. And when we use theory in that way, we're using theory in the same way that scientists talk about hypotheses. Hypotheses are proposed explanations for natural phenomena that are observed in the world. And because of how the scientific method works, hypotheses are not provable. In the scientific method, you can only disprove a hypothesis. And so the way that science operates and the way that scientists do their things is that they notice something and they propose hypotheses about that. They make educated guesses about the likely way in which that natural phenomenon is happening. And they make a bunch of them. And then they very systematically set out to disprove, to design experiments that will either disprove the hypothesis or not disprove it. And in this way, as one hypothesis after another is disproven, we are left with a hypothesis that cannot be disproven. And so when enough evidence builds to suggest that this hypothesis is actually very explanatory of whatever the natural phenomenon is, then that hypothesis or a set of hypotheses will be elevated to a theory. So in this really kind of confusing way, a theory is an unprovable explanation of how the natural world operates that is well supported by all the available evidence. So if contrary evidence turns up, then that theory has to be re-examined and altered to accommodate whatever this new contrary evidence is. So a theory then is a hypothesis that has been so well tested that it cannot be disproven and therefore is taken by scientists as the explanation for the natural world process that the theory describes. So a number of things that scientists consider to be facts: that all living organisms are composed of cells, that the genetic structure of populations changes over time based on natural selection and other processes, that is, the theory of evolution, by the way, that those are facts. These are scientific theories that have not been disproven. So again, bottom line, a theory is a law to a scientist. Okay, so meanwhile, back in microbiology, we're now in the mid-1800s, and up through this time, there were still many people who were arguing for the idea that non-living things could, through sort of, I won't use the word magic, but through unexplainable processes, would spontaneously become living. So this idea about how living things could be made from non-living material is called spontaneous generation, or abiogenesis. A meaning without, bio meaning life, and genesis meaning beginning or starting. So it's the idea of non-living stuff giving rise to living things. So it was believed, for instance, that moist grain could produce mice and rats. Well, we know that mice and rats are going to be drawn to grain, and so of course there would be lots of mice and rats hanging around a bag of grain. Or that mud produced worms and frogs, and we know those are the habitats in which worms and frogs lived. But to these early observers, it appeared that they just were springing up from the mud fully formed, or that meat left out could produce maggots and flies eventually. And this was all based on the idea that air had some vital force that could promote this spontaneous generation, that there was something in air that gave it special powers to cause non-living stuff to become living. Now, even though this was a held idea by many people, there were many who also did not think that this was the way that things had worked. And so for hundreds of years, scientists have been attempting to very conclusively disprove this idea. For instance, in 1665, an Italian monk conducted a couple of experiments. His name was Francesco. Oh my gosh, I've spelled his name wrong. Here, it should be an E. That's embarrassing. Francesco Redi. So Francesco Redi conducted these experiments around the idea that decaying meat could not produce maggots and flies. So he took 2 jars of decaying meat, he left one open and he left one sealed. The open jar got maggots on the meat. In the sealed jar, that should say no maggots. Dang, whoever my editor is, I've got to track down that dude and take him behind the woodshed. So in the sealed jar, there were no maggots that developed on the meat. But critics said, oh, Francesco, you know that air has this vital force, this living something that would have made maggots grow from the meat had the meat had access to it. So he designed a second experiment. Again, 2 jars with decaying meat. One was open, and one had a fine mesh covering all over it. In the open jar, where flies could visit, maggots grew. But in the netted jar, there were no maggots growing on the meat. What they noticed, though, was that there were maggots growing on the netting itself. So clearly flies were coming to the jar and laying their eggs that hatched out then on the netting, but they didn't have access to the food. So this was one of the earliest attempts that's documented to show that spontaneous generation didn't occur. So, okay, maybe it doesn't work for meat, but we know these other things, that if a hair from the tail of a horse falls into a puddle of water, then it's going to give rise to these Gordian worms, also called horsetail worms. Or horsehair worms. And so the debate raged for quite a while. Despite Redi's powerful evidence for a theory of biogenesis, that under current conditions, living organisms only arise from other living organisms. And this idea of biogenesis was proposed by a fellow scientist named Rudolf Virchow. He was publishing his work in 1855, but it was likely plagiarized as we look back on it now. But this just wasn't enough to convince people that in all cases spontaneous generation could not occur. So those arguments continued until 1859, when a French man named Louis Pasteur resolved the issue with a series of experiments that he did. So he devised some experiments that could prove that microbes that are present in the air, primarily on dust, could contaminate solutions that were sterile. But that it's not the air that's producing these microorganisms. So here we see a view of Louis Pasteur, a photo taken of him. He later became was in charge of what was called the Pasteur Institute. Lots of really important scientific work was done there. So here is Pasteur's experiment. First, he boiled meat in meat broth in a flask, and he boiled it long enough that it was sterile, or reasonably sterile. Then, using a flame, he heated the glass at the neck of the flask, and he drew that glass out into a long, curving, what's known as a swan neck flask. So here is one of his swan neck flasks, and here you can see this long, curving neck. And what he found, that when he set his swan neck flasks out with access to the open air, so air could certainly make its way up the swan neck, but if the air entered in, then any dust would settle out down here in this neck. So he had devised a very ingenious method of making sure that whatever dust and particles and microbes might be in the air would settle out before the air could make it to his to the broth itself. So what he found was that even years later, and there are at the Pasteur Institute, they still have some of his swan neck flasks, and they are just as fresh and fresh might not be the right word, but certainly uncontaminated as they were when Pasteur made them back in the late 1800s. So he showed that even with access to air, the broth would not become contaminated with microbial growth. And then to show that whatever was in the air had settled out in the crook there of the swan neck, he tilted his flasks so that fluid then ran up the flask and down into the swan neck, could grab now the dust and stuff, and then he tilted it back again so that everything ran back into the broth. And within hours, that broth would become cloudy with microbial growth. So what he was demonstrating, that there was no vital force in air, but instead there was dust with microbial life on it, in the form of spores likely, and it was the microbes that were causing the food to spoil or that were leading to the development of life, not the air and some vital force that could cause spontaneous generation of life from non-living stuff. So, Pasteur, though, it turns out, was really, really fortunate in his studies because other scientists were doing similar experiments to disprove spontaneous generation. But what they failed to take account in was the fact that endospores, these are very resistant life stages that are found inside bacteria, some bacteria, while they're alive, and then are resistant even after the bacteria die, remain able to hatch out, essentially, not really, but when the spores, they can dry out, they can withstand freezing and extreme heat and still do just fine. We have found these kinds of endospores that are more than 25 million years old, and they are still able to then germinate and produce living microbes. So Pasteur was lucky. He just happened to be working with broths and that as he sterilized them, there were none of these endospore-forming bacteria in them. Otherwise, he would have had microbial growth. And it turns out that so much of science rests on this kind of luck of just being in the right place at the right time. But I do like one thing that Pasteur says. He says that luck favors the prepared mind. So, Pasteur was fortunate and lucky, but he also had done a lot of work to make sure that he understood what he was up against. Okay, so that does it for this half of the history of microbiology, or a very brief history of microbiology. We'll pick it up in the next lecture. You know what to do, though. If you need me, need to check in on something, don't hesitate to email me or with your questions.